Hello and welcome to DNA Today. I'm your host, Keir Deneen. I'm informing you what is happening in the genetic world. The show's packed with news stories, lessons, debates, all to keep you updated and educated on what's happening in the world of genetics. And today in the world of genetics, I'm talking about gene therapy. So news stories are all centering on the concept of gene therapy. So before I get into all of that in our genetic roundup of the week, I want to explain what gene therapy is and the different methods of gene therapy. I also want to present both sides of the debate. Hopefully unbiased, that's my goal for today. Both sides of the debate whether this gene therapy is ethical and worth pursuing as a treatment for genetic diseases. And then we'll get into all the new research and studies coming out. So, gene therapy. We're talking about genes and proteins. So genes code for proteins, and when the genes are mutated, these proteins can be negatively affected. So these proteins can malfunction and cause genetic diseases. When there's an issue between the genes coding for proteins and the proteins not functioning right, that's when we may run into a genetic disease. And gene therapy is a relatively new technology that can prevent or treat or even cure diseases by correcting defective genes. And there's a few different approaches to gene therapies that doctors and researchers are all currently trying or researching, and we really are in the research phase for all of this. We're not really putting this more in humans that much. So the first one is replacing a mutated gene that causes the disease with a healthy copy of the gene. So you're kind of just swapping them out. The second one is inactivating a mutated gene that is functionally improperly, and that's known as knocking out a gene. I referred to this maybe last episode, 2 episodes ago, when I was talking about research presented at Jackson Labs for the grand opening there. One of the presenters was talking about how they knocked out a gene in a mouse and was successful with that. The research indicated that it did work. So that's the second way, knocking out a gene, inactivating that mutated gene. And the third method is inserting a new gene into the body that can help fight a disease. So this is the most common type. And this is where a new gene is introduced into the body. And this is usually through a vector, like a carrier. And it's used to deliver the gene to a specific targeted cell. And the most common type of vector is a virus. So first we hear viruses, that's bad and stuff. But really it's just a carrier. It's, the little package, we can think of it, of just delivering it to the site where the gene needs to be. And viruses can be genetically altered to carry human DNA. So like I said, we can put the DNA in and target it to a specific part of the body or organ or whatever. And once the vector sticks onto the cells, it can deposit the genetic information into the cell. And that's how that's working. So we got 3 methods going on. We got replacing mutated gene, second one is knocking out a gene, and the third one is just inserting a new gene. And a little history of what has happened in gene therapy is on September 14th of 1990, the first person with genetic disorder was cured with gene therapy. It was a 4-year-old girl suffering because her body didn't produce a crucial enzyme, ADA, which resulted in her having a weakened immune system. And the National Institute of Health Clinical Center in Bethesda, Maryland, took white blood cells from the girl and inserted the ADA-producing genes, so that would the genes that produce the ADA enzyme that she's missing, and then insert these genes into the girl. And as a result, the girl's immune system tolerance was greatly increased. So this was a very successful trial and everything. But there have been a few setbacks in gene therapy. The most major is in 1999 when an 18-year-old Jesse Gelsinger died after going through gene therapy trial, hoping to cure his OCTD, orthonine transcarboxylase deficiency. And his death was ultimately from multiple organ failures 4 days after starting the gene therapy, which is believed to have been triggered by a severe immune response to the gene therapy. And so it's very closely linked because he started it and then 4 days later he was having organ failures. So that was obviously negative. So we're having some successes and some failures. And another setback was in January 2003 when the Food and Drug Administration temporarily halted all gene therapy trials that use retroviral vectors in blood stem cells. And after a second child treated in French gene therapy trial had developed leukemia-like condition, and a few months later in April 2003, the ban was lifted. So it was really small time frame that Food and Drug Administration had halted all the gene therapy trials, but it still is part of the history of genetic therapy and how we're evolving with that and more experience with that. And because scientists and doctors are still learning how to perform gene therapy, there are many risks involved in the process, and it's only used if the disease has no other known cure. However, in the future, these processes could possibly replace drugs and surgery, because the more we learn about it, it could end up being the more efficient way and have more successes than drugs and surgery. And it maybe even would have less side effects if you're really efficient with putting it in and not having side effects for it, whereas drugs and surgeries can also, recovery time for surgeries, different side effects of drugs. So it could really, it could advance things and in terms of treating genetic diseases. And currently, the FDA has not approved any human gene therapy products for sale. So I want to emphasize that we're still in the experimental phase of all of this. So I want to kind of get into the pros and cons of gene therapy and what's out there for what people are believing, what the two arguments are. So I'm going to start with the pros of genetic therapy, gene therapy. And many countries have been approved for trials in gene therapy. That's not to say that they're approved for products. This is just trials. And these countries include the USA, China, France, Italy, and the Netherlands. And it has an incredible therapeutic potential. Many genetic disorders have no cure unless the defective gene is replaced by the correct one, and this is precisely what gene therapy aims at, is replacing that gene, either by knocking it out and silencing it so it's not operating anymore and producing proteins, or that you're replacing the gene, you're actually taking it out and putting a new one in, or just introducing a new one that maybe would be more dominant over the other gene. And so if certain genetic diseases can only be cured, treated by genetic therapy, this poses the argument that shouldn't the public have a right to these possible cures when there's no other known cure for these diseases, like Parkinson's or Huntington's that we've talked about previously on the show? That does the public have a right for research to be going on and have access to genetic therapy if the potential is that it could be working? Another point is that genetic therapy not only can benefit those affected by genetic disorders, but also those that could inherit the disease. If gene therapy can target reproductive cells of carriers of genetic disorders such as cystic fibrosis, Parkinson's again, or even cancer, it's possible that children of the carrier would be free of the defective gene. So if cells were reproductive cells, eggs and sperm, were able to see if the genes were there and maybe knock them out in that first sperm or egg cell, then the organism wouldn't have that disease, because if you, say, silence the gene for cystic fibrosis, then cystic fibrosis wouldn't occur. So if we're actually able to do this, it's possible that any children of the carrier would be free of the defective gene. And thinking of this on a big picture of, hundreds of years down the road, it would really start the prevention of genetic diseases and passing on genetic diseases. So if by some, science miracle that 100% of these could be effective, then, hypothetically speaking, this could be eradicated, that genetic diseases, straightforward genetic diseases at least, wouldn't occur anymore. And when successful, gene therapy has loads of advantages over drug therapy, as I mentioned before. This includes providing a cure rather than just easing symptoms. This is something I mention a lot. I like using the example of cystic fibrosis because it is very straightforward, and it's one of the more common genetic diseases. And cystic fibrosis causes sticky, thick mucus to build up in the lungs as well as other parts of the body. And instead of just treating the buildup of the mucus through techniques that trigger strong coughs to help loosen and clear the mucus, of getting rid of it, the symptoms could possibly be eliminated, that instead of just getting rid of it in terms of after it's being produced, let's just stop it from being produced. And that's something that gene therapy could possibly accomplish if the research is advancing as well as the technologies. So that's kind of the pros and all of the highlights of why people are interested in gene therapy. But now we'll kind of look at why people are against it. What are the cons? Obviously, something that usually comes to mind when these things are talked about is the expense. Gene therapy treatment will be expensive, and more research needed means more funding. And of course, the question is, where's the money coming from? The cost of gene therapy is high at this time currently, and could be a concern for families that need a lifelong treatment and don't have health care. So you get into the issue of that people that have more money can afford these things, whereas people that don't have as much money can't afford these things. But just like other things in genetics and science in general and technology, over time in the long run, these things will be cheaper. So it's really just a matter of time before technology advances so much that things are much easier to do and straightforward and I mean, the possibilities are really, really advantageous that it can eliminate suffering of the individual and possibly their children. Again, as we were saying, that if the gene could be silenced or knocked out or something, that then an offspring couldn't have it. But really, the point here for the con is that the cost is going to be great, and will only certain people be able to access this, and other people will not. Another point is that many people with strong religious beliefs reject the ideas of manipulating the genetic makeup of humans. So the mindset here is that altering genes is similar to tinkering with nature or messing with what God had set up. And so there, I'll get into something else too later that talks about this a little bit. But yeah, so this people that are very religious that don't believe in manipulating the genetic makeup of humans are against this idea of, because you're changing your genes, you're changing what codes you. So you really are changing yourself on a molecular level. And there's no guarantee that the vector carrying the healthy gene will end up in the specific place it's intended. So if it's targeted at the liver, it's hard to actually say that it will get there depending on the method used to actually target that organ. And there's a risk of causing even more damage to the genetic makeup that can result in severe consequences for the patient. Like I mentioned before, that 18-year-old kid that died after 4 days, he started showing symptoms 4 days after starting the genetic therapy. So that obviously did not work, and that was a failure. But it's consequences like these that there's no guarantee in science, but in here, you're really you're messing with people's lives. So there's a huge, huge disadvantage and risk of patient dying and having severe consequences. And I mentioned this a little before of couples looking to have kids. If they're aware of their genetic makeup and know that they're both carriers of a specific genetic disorder, they can make a more informed decision of having children. However, this may not, this could lead to things such as genetic screening. So you can screen to see if the embryo, say if they've already conceived, if the embryo or fetus has a genetic disease, and the couple may decide from there if they want to continue with the pregnancy or not. So this could be a positive or a negative. I kind of just ended up throwing it in this category, but it really can be seen as a positive or negative depending on your view of abortion. And going along with this idea is the fear of eugenics. And this is a term that denotes the creation of the superior race, and an idea that the media has tried to embody in the concept of designer babies. So designer babies is what I was hinting at before, and basically designer babies are where specific genes are selected in order to create the perfect child, and this concept is often compared to Hitler's attempts to create a superior race. So things such as genetic screening and genetic therapy could be taken too far in some people's eyes, that couples could say, I want a kid with blonde hair, blue eyes, and they select genes, and similar to the movie Gattaca, which is awesome. You should watch it this weekend. It's on Netflix. But anyway, this concept of designer babies is a scary thought, because then we'd actually start designing our human race. And so, as a lot of issues in science, a technology or concept can be taken too far, and there's always that risk. But those are kind of the two main sides of what genetic therapy is all about. So now getting into what the news is surrounding genetic therapy, now that we have a better idea of what it is and the consequences of it, and possibly the advantages of it. So our first news story is that a gene mutation may lead to the development of new cancer drugs. Scientists at the University of Michigan and Health System have discovered a gene mutation that causes rare premature aging disease called dyskeratosis congenita, or DC for short, which I'll call it. And this disease affects stem cells so that they can't regenerate. And people affected by DC age prematurely and are prone to cancer and bone marrow failure. And this mutation could lead to the development of drugs that would prevent the current uncontrollable cell division that causes cancer. And J.K. Nandakumar, the assistant professor in the Department of Molecular, Cellular, and Developmental Biology, explains that this study has found research that could ultimately lead to developing new drugs that prevent cancer from spreading. So previously on the show, I've mentioned telomeres, or telomeres, however you want to say it, which are the end parts of the chromosome that protect the chromosome from being degraded. And telomeres are also linked with aging, and we think the shortening of telomeres throughout an individual's life has an impact on aging. So how does DC fit in all this? So the mutation that causes DC occurs in a protein called TPP1, And this mutation inhibits TPP1's ability to bind the enzyme telomerase to the ends of chromosomes. And as I said before, telomeres help stabilize the cell, and without them, the DNA is not protected. It's kind of, I explained it like a shoelace, and that end part, that plastic part called the aglet on your shoelace, it's kind of like that. It's preventing your shoelace becoming all frayed and prone to everything you're walking through. And it helps stabilize the cell, and without the telomeres, DNA is not protected, and therefore less cells are dividing, and it's messing with the cell cycle. And these patients have low levels of protein that binds to the telomeres in their chromosomes. And again, this telomerase is what binds telomeres to the ends of the chromosomes. So now that we know kind of that's what's occurring in DC, the telomerase is not working properly, how does it relate to cancer? Well, cancer works in the opposite way. So cancer patients often have an abundance of telomerase, and this causes cells to divide with less control. It's out of control. And the assistant professor I mentioned before explains this concept that, quote, telomerase overproduction in cancer cells helps them divide uncontrollably, which is a hallmark of all cancers. Inhibiting telomerase will be an effective way to kill cancer cells. And he also explained that gene therapies could be used on DC patients to repair the mutation that causes telomerase to not be produced, causing telomeres to not be attached properly. So gene therapies could also be used for the opposite effect on cancer patients, where a drug could inhibit telomerase, which is overactive in cancer patients. So we're increasing telomerase in DC patients, whereas we're lowering it in cancer patients, and hopefully both of those would end up putting people in the normal zone and preventing cancer as well as DC. Now, the first step towards these two goals is to experiment with DC cells in the lab and see if it's possible to fix the TPP1 mutation so that they, just like they're predicting, so that then telomerase would start being properly produced, as well as the cell division to be stimulated in stem cells of the DC patients, that if they're correct in this association between the TPP1 mutation and the amount of telomerase, then hopefully this study of this would prove their point and also be able to fix this. And J.K. Nandu Kumar also shared that he thinks it's, quote, conceivable that with the recent advancement in human genome editing technology, we could, in the not-so-distant future, repair the mutation in hematopoietic stem cells in the bone marrow of DC patients. That's, again, where the site of the problem is, really the heart of it. And this genetic disease is a great example of how one small change, just one mutation, can cause detrimental effects on one's health. It really can. You think about all the genes in our body, it only takes one to be messed up to, be lethal. And Nanda Kumar also reflected on this concept, saying, quote, it was surprising to us that just deleting one single amino acid, so like amino acid, that's only a codon, so that's like three base pairs. But by going back to the quote, just deleting one single amino acid and protein chain that is 544 amino acids long can result in such severe diseases. And this is a concept that is in a lot of news stories and a lot of academic journals, that we're as stable as DNA is and how flexible the molecule is in terms of changing and a lot of things happening to it, and it really is coding for us. It only takes one mistake to make a huge detrimental effect on the human body and as well as other organisms. Obviously, we're more focused on humans for this news story, but it's definitely a big problem. And so going into the next news story, I mentioned gene therapy now, and that in terms of one mutation causing low levels of telomerase in DC patients. And as I keep mentioning, gene therapy is still in the experimental world. It's not being used as a treatment quite yet on the general public. But new research also has more promise that this method of treating genetic diseases may be possible in terms of the mainstream. Now, long-term treatment success using gene therapy to correct a lethal metabolic disorder has been reached. And this study I'm referring to focuses on the mutations which cause an excess amount of bilirubin and in the blood, and to the point where it is lethal. It has so much of this bilirubin that it is lethal to the organism that is affected by this. And bilirubin is the product of a part of hemoglobin, which is found in the blood, and a part of hemoglobin that's broken down and bilirubin is excreted into bile and urine. And this mutation occurs in a single gene. Again, I'm talking about one gene. I like focusing on this because it's showing that gene therapy can make a difference when it's one gene, and then hopefully down the road, if we can master this genetic therapy of one gene, we can then do it for multiple gene disorders. But again, this mutation occurs in a single gene, just like, again, the mutation of DC cells are believed to have. And this single mutation causes excess bilirubin, and the metabolic disease with this is known as Crigler-Najjar syndrome type 1, also referred to as CNS1 for short, which I'll be referring to. And the study was able to correct this mutation in CNS1 in mice, which is very often used in genetic studies, and the researchers achieved significant long-lasting results in the bilirubin levels, which means they were able to target the gene correctly and fix it so it produced a healthy amount of bilirubin instead of excess to the point of being lethal. And this research was published in the Human Gene Therapy, so you can read the full original text there. And the head author, Julia Borloo-Tulisi, shares along with co-authors from the International Center for Genetic Engineering and Biotechnology, along with a lot of other institutions. That the list was quite large. But just how effective was this gene therapy? They said, it worked and it was long-lasting, but what's the breakdown here? So there was 70 to 80% reductions in bilirubin levels early among treated animals, and later 50%. So it really, the studies are finding that this really is working. And the gene therapy had two delivery strategies in this study. The first was targeting the therapeutic gene directly to the liver, or preferably the skeletal muscle. That was their aim, to really be looking right at the liver skeletal muscle. That was what they were doing. And Dr. James M. Wilson, the editor-in-chief of the Human Gene Therapy, where this research was published, again, you can find it online, he commented on the significance of these findings, saying that, quote, CNS1 is an outstanding model for in vivo gene therapy with easily measured and clinically relevant metabolic endpoints. So these stories are really significant in the promise of gene therapy really helping, really making a difference. And the more studies that are coming out, the more we build upon this base of learning about gene therapy, we may actually be able to use this in the not so distant future on more of those straightforward Mendelian genetic disorders, such as cystic fibrosis, Huntington's disease. And these are disorders that affect a large number of people, and being able to fix this would be really significant. So it just goes to show that gene therapy does have a lot of promise, but there are, I'm highlighting a lot of the exciting news stories of what's developing and the research that's developing, but there are a lot of consequences that need to be considered when using this, and a lot of ethical consequences too, and just making sure that we don't go too far with it. And the question is, then what is too far? But in terms of this, it's definitely exciting to see that these researchers are finding answers and are really expanding our base of knowledge for gene therapy. So I'm sure I'll find other gene therapy stories to throw into further episodes, and I'll definitely keep you updated, and I'll try and choose those over other news stories because it's hard to pack it all in. But that's the show for today. That wraps up the episode for DNA Today. You can tune in next Monday at 1 p.m. and every Monday at 1 p.m. to learn more about genetics on DNA Today here at WHUS Radio. You can also go to DNApodcast.com. I've got a lot of great resources on there and other links to a little bit bigger institutions of things of what they're doing and learning more on there. There's so much to learn online, especially with science and keeping up to date on things. There's only so much I can go over in half an hour, so I highly encourage you to go on and check out news stories and even try and read some of the original publications. It can be a little hard to get through, but if you do have some experience in science classes, it definitely is doable. You can follow on Twitter at DNA Podcast. You can follow me on Twitter personally at KIRADINEEN. That's Keir Dineen. And if you have any questions, shoot me an email. I'm at info@dnapodcast.com. I'll love hearing back from you, even if you just shoot me an email, say, hey, love the show. It'd be cool if you covered more of this or that. It's always good getting feedback because without feedback, it's kind of a one-way thing. So definitely be, you can even reach out on Twitter or the Email address, info@dnapodcast.com. Also, there's a contact form on the website, dnapodcast.com. Can't say it enough. So thanks for listening, and you can join me next week, Monday at 1 p.m., and every Monday at 1 p.m. to learn and discover new advances in the world of genetics.